Hello and welcome to this lecture on morphometry as part of the ENIGMA-U lecture series. Thanks a lot to Emily Dennis for organizing this lecture and inviting me. My name is Philipp Sermann. I'm working at the Max Planck Institute of Psychiatry, Munich, and I'm a clinician and an imager. Let's start with a general definition of morphometry before we come to the basic chapters of this lecture. Morphometry is the quantitative measurement and analysis of the shape, size, and structure of objects or organisms. However, in the context of ENIGMA and neuroscience, the term mainly refers to brain morphometry or morphometry of the central nervous system, because the spinal cord is also part of the CNS. Morphometry is a subdomain of neuroimaging, and as you may know, the main source of images that we use for morphometry comes from MRI. So I also refer to the MRI physics lectures that help you to understand the acquisition of MRI images. Generally, morphometry is a scale-independent principle, and its exact meaning is context-dependent. So it may be applied to the whole brain, but it can also be used for smaller scales such as histology or even single-cell structures. Cross-species analysis are also very important and very informative, for example, on evolutionary aspects. On the left side, you can see a post-mortem brain specimen, and you can already detect very typical features of a brain. You can see its gyral structure, you can see sulci, you can see the larger fissures between the gyri and the left and right hemisphere, for example. You can see there are moments of asymmetry between the hemispheres. You can distinguish the larger upper part, the cerebrum, and the lower, smaller part, the cerebellum. So when anatomists or pathologists dissect such a brain, they try to create orthogonal slices with a parallel slicing direction. So that would allow for a optimal standardization of the diagnostic process. And it also makes the slices comparable between individuals to a certain degree. The whole procedure, of course, demands enormous knowledge of landmarks and preparation, but the whole process, getting information in slices, is very similar to what we are doing in neuroimaging. Now, the most simple measurement of the brain, I would say, is its total weight. If you ask yourself, is this measure, the total weight, correlated with what we actually would like to know, the content of the neuronal system, then you will see there is some discorrelation. So generally, smaller mammals also have smaller brains, and to a certain degree, this correlates with the neuron count. But there are also exceptions. If you look at the scale below, you will see that the African bush elephant, for example, has a brain weight that is more than double that of humans, but its neuron count is much lower than that of humans. So there must be sources for this discorrelation. For example, you might have different folding patterns, different packing efficacy, and also different proportions of anatomical connectivity and fiber systems. So yes, across species, we could say that more neurons correlate with intelligence, but within the human species, this is much more complex. So if you're interested in this interesting question, is there a correlation between the number of brain cells and IQ? I refer you to this very interesting paper. Now, if you look at one brain slice in more detail, one can immediately see it is mainly consisting of two intensity or color compartments, the more beige grayish part and the white part. The gray color comes from a high density of somas of neurons. Um, and of course, to a certain degree, the color is also caused by the postmortem preparation. One can easily see there are different compartments that contain gray matter. It's the cortex or the cortex mantle, and there's also more deeper gray matter structures referred to as the subcortical gray matter, and not shown on this image, there is also a cortex of the cerebellum, of course. The white matter contains millions of axons, and its color mainly stems from the high lipid content that is needed for the electrical insulation of the axons. It should also be mentioned that the complementary cerebrospinal fluid space, both the outer or external CSF space and the internal CSF spaces, are quite important for morphometric techniques, particularly computational morphometry, because it also gives a sharp contrast to white matter and gray matter. There are also advanced tools that can segmentate out more than these 2 or 3 compartments, for example, blood vessels and meninges. Now, gray matter does not exclusively contain neurons, but it also contains axonal tracts. It contains glial cells, capillary vessels, and of course, the dendrites, so the whole dendrite tree of a neuron. There's also a term called neuropil that is defined as a mix of dendrites, unmyelinated axons, and glia. White matter also does not only contain the mentioned myelinated axons, but also unmyelinated axons and glial cells such as oligodendrocytes and astrocytes. The same basic 2-compartment structure can also be found in the spinal cord, but we will not go into the detail of the anatomy at this point. One of the earliest slicing techniques that has been developed in radiology is computer tomography. Computer tomography needs X-rays that are sent out and then passing through the specimen or the body and then detected by a detector ring. Modern CT systems have a continuous forward movement of the specimen in a kind of spiral technique. And by analyzing the collected signals, these systems can then provide you with slices of the specimen. And what's important to know about the CT technique as the historical technique when it comes to morphometry, but still very important for the clinic, of course, is that the whole information content gained from CT is correlated with the tissue density. Here you can see that CT gives you a very good contrast between the CSF spaces and the brain, which, for example, is very important if you are analyzing an emergency patient looking for shifts of the CSF spaces or swelling of the brain, for example. You also see air quite well contrasted and bone. However, the contrast between gray matter and white matter is not very good. MRI provides you with a much better contrast between gray matter and white matter. And this, as we will see later, is absolutely elemental for morphometry. It also gives you a good contrast between gray matter and CSF and white matter and CSF. Um, as you might know, MRI allows you to fine-tune your sequences to specific tissue properties, so it's much more flexible in terms of the uh, information content that you can generate from such images. Here's another example of a sagittal image acquisition. Um, it's now scrolling through the brain, similar as a radiologist would analyze this brain in a clinical case. And you can nicely see the gyral structure. You can detect the cerebellum, the CSF spaces, spinal cord. The next chapter is actually very brief. It will take you from the early era of CT to computational morphometry. Radiologists like to perform quantitative measurements, particularly distances and volumes, because they can be used in an objective way to build up databases, to generate normative scales, and so on. So on the left side, you can see an example of a Huntington's disease patient. This disease is characterized by a specific loss of volume starting in the caudate nucleus. And by calculating a ratio between the distance between these caudate nuclei and the edges of the frontal ventricular spaces, one can estimate if there's a specific volume loss of the caudate nuclei. So this is a simple technique, and it's quite sensitive to at least the more advanced stages of the Huntington's disease. In the mid column, you can see an example of what's called progressive supranuclear palsy, a very rare Parkinson's-like disease in which the upper brain stem, that is marked by an arrow here, looks a bit like a hummingbird. So it can also be very complex features that the radiologist is looking for. Also, the midbrain diameter is reduced in this disease. On the right side, you see a more common disease, Alzheimer's disease, in image B compared to a normal control person in image A. And what are typical features of Alzheimer's disease you could measure as a radiologist? It would be a widening of the temporal horn, so the CSF space next to the hippocampus. It would be the height of the hippocampus, and it would be the width of the choroidal fissure that is also neighbored to the hippocampus. This takes us to a very commonly used technique called manual morphometry. Manual morphometry basically means that you circumscribe certain brain structures guided by landmarks and a lot of anatomical knowledge and rules. And eventually, you would either count a voxel in or out your structure, and by this gain a simple volume value. This technique has been extensively used, for example, for measurements of the hippocampal volume. You can also measure pathological spots of tissue, for example, lesions that are standing out bright here on this FLAIR image. There are both manual techniques and automated techniques. You can compare these techniques by, for example, using the DICE index. The DICE index is a classical measure of inter-methods reliability. It's very easy to calculate. You double the area or volume of the overlap and divide it by the sum of both areas of both techniques. The closer it is to 1, the more precise the overlap between the techniques is. There are now very advanced automated segmentation techniques, for example, to detect subregions of the hippocampus. They are computationally more extensive. They are mainly automated. They require either one channel or several sequence channels as an input. And in the background of these algorithms, you usually have a lot of training data coming from, for example, post-mortem MRI with an excellent resolution, also supported by in vivo training data. And this system then generates a kind of statistical atlas that is then warped to the actual data that you offer the algorithm. And by this Bayesian segmentation logic, you can then estimate these subregional volumes. The same principle is meanwhile available for the whole brain in a very new version of the FreeSurfer software. Here also um, high resolution postmortem MRI information has been fused with in vivo segmentations and allows you to gain histologically or quasi-histologically informed um, parcellations of the brain in a fully automated manner. Now, in this next chapter, I would like to explain why brain segmentation is a key step for computational anatomy. As you've seen in the postmortem brain specimen, it's obvious that there are at least three microscopically detectable tissue classes. So the goal of a segmentation program that processes MR images is to at least find these three classes in a reliable way. Um, the outcome can be a hard segmentation, so a clear class, gray matter, white matter, or CSF per voxel, a kind of the winner takes it all strategy. But it can also be a soft segmentation, and here rather a tissue probability is the result. So the likely class of a voxel in the original image doubtlessly mainly depends on its intensity. But it also depends on the location if you have the image in a, say, standardized position. For example, a voxel that is likely to be located in white matter or in a typical position of white matter is more likely to be white matter than gray matter. And it's these two channels of information that the segmentation programs tend to use, the location and the intensity. If you look at the typical intensity distribution of three validated tissue classes of a classical T1 weighted image, you will see that the distributions overlap. And it's this overlap that causes problems because you want to make a reliable decision on the class of that voxel. So this is where the location information comes into play. On this example of three different scanner systems, you can also see that there is absolute biases, intensity biases, depending on the scanner platform. And this is also a problem for such algorithms because you cannot use absolute intensity cutoffs, but you have to build an algorithm that analyzes these intensity histograms on a more relative basis. And naturally, you would expect the same segmentation result if you have scanned the person on three different scanner platforms.